Thank you. Okay, so thank you everyone for being here, and thank you to the organizers for having invited me to really start this section. My name is Juan Vizcaino. I'm based at the EBI, European Bioinformatics Institute, and the title of my talk is Write on ProteomeXchange, Making Proteomics Data Fair. So this is the overview of my talk. I'm going to first describe the current status of proteomics data repositories, Write on ProteomeXchange. Then I'm going to talk a little bit about Proteomics Standards Initiative. Then I'm going to talk about data reuse of public proteomics datasets, and I will also finish my talk about talking about future challenges. But before starting, I thought that it was really a good idea to start with a one-slide introduction to proteomics, and it's I think key to say here that it's increasingly relevant in the life sciences for many possible use cases: protein expression information, proteins in time and space, protein-protein interactions, post-translational modifications like phosphorylation, protein localization, dynamics and turnover, also structural biology, multi-omics approaches. Really, proteomics is increasingly relevant in the life sciences. Mass spectrometry has always been the main workforce of proteomics, but it still is, but it's worth highlighting that in the last few years, other methods are becoming increasingly popular, like antibody-based techniques or the use of other affinity reagents like Olink and SomaLogic platforms. So my team at the EBI is responsible for PRIDE, it stands for Proteomics Identifications Database, which is the EBI resource that stores mass spectrometry-based proteomics datasets. So far, we have only stored mass spec data coming from all types of approaches. Is by far the world-leading resource, storing more than, at present, 34,500 datasets. And it's important to highlight that since a few years ago, PRIDE is an ELIXIR core data resource and data deposition database. And I think what I would like to highlight here is that what we did very well in the proteomics community, let me say this at least, is that we really work together. This hasn't happened in other kind of sister fields like transcriptomics or metabolomics. And we started working together more than 10 years ago, establishing a global infrastructure to implement standard data submission and data dissemination practices between the main proteomics repositories. So ProteomeXchange was started in 2011, also under the leadership of Henning, who is here in the audience. And PRIDE is the European resource, but then there are 3 resources at the US, and some of them are funded by the NIH. So we have PETA Dallas in Seattle, Massive in San Diego, and Panorama also in Seattle. And then we have also 2 resources in Asia, IPROX in China and JPOS in Japan. So this gives us a very nice geographical distribution. And thanks to the perceived reliability of ProteomeXchange, but also thanks to the push of journals and funders, really data sharing has generalized in the proteomics field in the last few years. Now, I mean, the status of data, the open science practice in proteomics is kind of analogous to what happens in transcriptomics, just to put an example. And to recognize the importance, ProteomeXchange was named a global core biodata resource at the end of last year. So, of course, in this first part of the talk, I'm going to focus mainly on the findable and accessible aspects of the FAIR data principles. And this is what we are doing in ProteomeXchange, how it works. Basically, the first stable data workflow was established in 2013. It has been refined since, also by the addition of new members. There is a common dataset identifier that is used across all resources. The datasets are mostly not replicated, so data remains in the original repository and doesn't move from there. There is a consortium agreement that I think is important. If one resource runs out of funding, what can happen? It has happened in the past. The others agree to take the data, so of course the community benefits. There are different types of resources, so there is a space for kind of generalistic resources like PRIDE, but also for smaller resources that are specialized in one particular technique. For instance, PETA Dallas Puzzle and Panorama Public are focused on targeted proteomics approaches. And there are common submission guidelines that were kind of agreed, again, many years ago. So for each data set, it is mandatory to have the raw data, the primary data coming from the mass spectrometer, but also the process results, identification and quantification, minimum set of experimental and technical metadata, and there is also, of course, a lot of additional information that can be optional. I'm not going to get into the details here, but it is important that users, now they have really a very good understanding of what a ProteomeXchange dataset is. Every resource may implement submission in a slightly different way, or they might have they can have their particularities, but it's really homogeneous from external perspective, I think. Also, data sets are private by default, and once the corresponding publication is out, the data sets become public and searchable in one portal that is called Proteome Central, that basically enables to search for any kind of ProteomeXchange data sets that are public. Here, you cannot look for private, obviously. You can only look for public ones. Okay, so I'm going to give some kind of statistics. I'm going to give them just from pride, because again, it's by far the leading resource, and also because it's easier for me to get them. So this chart shows the increase of data submissions year per year from 2010 to 2022. This is not cumulative. This is really a year per year. And you can see the amount of data in the public domain really continues to increase. In the last year, we had almost 6,200 data sets, which is around 514 submitted data sets per month. This is only PRIDE. Of course, there are other resources. PRIDE accounts for approximately 82% of the data sets. As I said, the world-leading resource by far. Of course, this has been possible due to huge improvements in infrastructure with a focus on stability, reliability, and automation. Another kind of column of all this kind of infrastructure is the Proteomics Standards Initiative. And this is important from the point of view of interoperability between databases, interoperability between tools and databases. And this was started even before ProteomeXchange, so around 21 years ago now, in 2002. The Proteomics Standards Initiative is the organization under UPO, the Human Proteome Organization, that develops open data standards for proteomics. It develops data standards, but also all the control vocabularies that are used by the different resources. It has different working groups. And also maybe for you to can be highlighted that there is really a closer interaction with the metabolomics community, because of course metabolomics has mass spec as the main kind of workhorse as well. What is important to highlight here is that ProteomeXchange resources are really closely involved in PSI activities. We work, many of us has like two hats, ProteomeXchange and PSI, because this is really key in order to improve the sustainability of the resources in the medium long term. This is the kind of summary of the current standards that have been implemented by the PSI in the mass spec world. Some of them, as usual, are more successful and more adopted than others. Maybe the one that can be highlighted a little bit more is MCML, which is the output from the mass spectrometers, and it is used also for, of course, for proteomics, but also for metabolomics and for other types of omics that use mass spec. But maybe let me highlight two of the most recent cases, just again to show this interaction that is key. One is the development of the universal spectrum identifiers, which is they provide a unique identifier for mass spectra in ProteomeXchange resources. So now it is possible to highlight any individual mass spectrum in one of the ProteomeXchange resources. So this really helps to make mass spectra data fair, and because of this, it's possible to access individual mass spectra and also PSNs using this universal identifier. This is the implementation in PRIDE, but every of the resources implements USIs in a different way. And also, let me highlight also a data standard for metadata and experimental design. It was lacking in the field for a long time, and we developed this 2 years ago. MageStaff proteomics, following the steps of our colleagues in the ABI, working in the functional genomics teams, ArrayExpress and Expression Atlas. And the goal here was to enable sample metadata annotation for public proteomics data sets, capturing the experimental design. So this is really essential work for improving reuse of data, and I'm really happy to say that this standard is increasingly adopted in the field. Okay, so I have talked about, the proteomics data repositories, PRIDE and ProteomeXchange. I have talked about the aspect of interoperability in the context of the Proteomics Standards Initiative. And now is my turn to talk about giving some highlights of data reuse of data sets. And this is really one of our main activities lately, because they are the ones that are easier to get funding for, because many of these activities are really quite novel. So again, concentrated on the reusable aspect, because now there is so much data in the public domain in proteomics I know this has happened in transcriptomics, for instance, for many years, but in proteomics is only relatively recent. So really, data reuse is flourishing, and it's flourishing for many different kind of use case and different approaches. And it's really, new papers get published every week. Of course, I don't have time to go through all these examples, but at least I wanted to highlight two of them. One is data reanalysis with different purposes. And in this case, I would like to highlight the use case of providing a consistent view on the proteome. So the HUPO, the Human Protein Organization, launched the Human Protein Project around 10 years ago with the objective of at least characterize or detect one protein or one proteoform for each human gene. So basically what happens here is that two of our ProteomeXchange colleagues, Vizcaino and Massive, they go to ProteomeXchange data sets, they download the ones that they think that they are more interesting, and then they reanalyze it using a consistent pipeline. And then they provide a consistent view on the proteome. The current view from last year is that this has been evidence found for approximately 93% of the proteome. So this is activity that it continues. Of course, the kind of most difficult proteins remain to be detected, and now maybe the focus is going to be increasingly put in actually the proteoforms. So proteoforms are the kind of different kind of protein species that can arise from the same gene. And a second example that I wanted to highlight is the increasing use of artificial intelligence, machine learning, deep learning approaches. And because there's so much data now in the public domain, this is something that can happen. This is a nice figure from a paper from Matthias Mann from a couple of years ago that summarizes the development of tools based on these approaches that can be applied to different kind of stages in the proteomics data workflow. So the ones that have a highest impact are the prediction of mass spectra, MSMS spectra, and this can be used for different purposes, for instance, to rescore the different tools, and this has really have a huge impact, and also the prediction of the retention time. But again, tools have been developed with many different use cases in mind, and really this has also been possible due to the high availability of public data sets, because many of these data sets have, of course, been used as training or demonstration sets. But maybe what is more interesting for a kind of more generalistic audience is that these kind of approaches are increasingly used downstream. So once you have the proteomics results, it's really interesting to use these approaches to improve the biological conclusions, to improve the biological knowledge. And I just highlighted here 3 papers that have been published in the last few years, applying to different proteomics subdomains like phosphoproteomics, metaproteomics, and metagenomics, or integration the metaomics approaches, or immune peptidomics. And this is really I think the use cases that are kind of most interesting for the field, because it can also be a kind of opening door for other people working on other fields that join proteomics. So I just wanted also to give some kind of idea about the scale of operations in Pride from the downloads and data reuse activities. So in the last 12 months, more than 1.1 petabytes were downloaded, coming from more than 184,000 unique hosts. And Pride datasets have been reusing at least 600 publications, which is, again, an underestimation, because, of course, these are the ones that we can detect, manually. And in the case of kind of what we are trying to do, again, involving many of these sources, is trying to bring proteomics data closer to life scientists in many different domains, in protein expression, post-translational modifications, proteogenomics, protein structural biology. And let me highlight just one case. This is in the case of post-translational modifications with our colleagues in UniProt. We are carrying out the ProteomeXchange project with the idea of making post-translational modification data fair. Again, is it the same concept? One takes data sets in the public domain that are suitable for translational modifications. They get reanalyzed using a consistent pipeline, and then the data is integrated into UniProt. And there are just also now some examples of some builds that are being integrated into UniProt this way, where the evidence for a particular PTN, in this case phosphorylation, can be traced back to the originating mass spectrum in one of the ProteomeXchange resources. Okay, so just the last minute for me to highlight future challenges that are upcoming. So the first one is actually one that has already been faced by our colleagues in other fields. So I know Mallory will talk about it later. So far, all the data in proteomics has been completely open, no restrictions. So one publishes a paper, the data gets immediately made available for you to download. And both Bright and ProteomeXchange are completely open proteomics data repositories. But what is going to happen, and it's starting to happen now, is there are some data sets that are being used in clinical research, for instance, that are considered to be sensitive. And they're considered to be sensitive for different reasons, because it can contribute to the identifiability of individuals, because of patient-informed consent reasons, or because of other kind of legal regulations, like the GDPR. So there is a lot of work to do here. Also, the funders need to realize that this work needs to happen. We worked a white paper about the topic a couple of years ago that's still, very up to date, when we made some recommendations of some developments that are needed related to policy making, bioinformatics infrastructure, involvement of proteomics in GA4GH. So, and of course, there is the need to have control access proteomics data repositories. And some other challenges, well, the increase in data, the data sets are becoming larger and larger, improvements in dataset metadata provision, or the storing of new data types that are non-MS. That's it. Conclusions. ProteomeXchange and Pride in particular have generalized data sharing in the public domain in the proteomics field. ProteomeXchange and PSI are two very nice examples of international, across-continent open collaboration. I think I'm really proud that we managed to do this in proteomics. As a consequence, multiple data reuse activities are being increasingly developed in the field, but of course, there's a lot of work to do still. So just to finish, acknowledgement to all the resources, all ProteomeXchange funders, and of course, especially to the Pride team. Thank you very much for your attention.